Excellent. Now, okay, so it's my pleasure to welcome both Jennifer Doubt and Eric SNYDER, who are going to speak on the bryophytes of Prince Edward County. So Sheila, will you take over and do the introduction, please?
Sure. Thanks, Amy. We're very fortunate and very excited to have two excellent speakers for this evening to tell us about bryophytes. Not too many of us know about the mosses and liverworts that we see. We see them all the time, but we don't take the time and effort to maybe identify them. And this evening will hopefully help us get on the right track to do that. As some of the plants, this is Jennifer kind of gave me a little description of what the plan for the presentation would be. And it's as some of the first plants out of the gate when snow and ice melt away, bryophytes make a fun quarry for nature lovers on early season hikes or even on an errand run in town. We'll have a chance to learn what sets mosses and liverworts apart from each other, from other plants, and from lichens, and to put names to some of the familiar Prince Edward County species. And we'll find out some of the many reasons it's fun and rewarding to discover bryophytes and take away some ideas and resources to help get us started doing that. So I'm going to go roundabout. Usually it's ladies first, but we'll start with Eric SNYDER. He sent me information about him originating in the Eco District 6E15, which sort of gives a general idea, but not exactly a specific location for where he grew up, somewhere between Trenton and Kingston. So maybe we'll find that out later on. Some of the members know Eric from his time in Kempville, when he discovered a population of pygmy mole crickets. Probably not many of us have seen these obscure burrowing crickets, and many of us may not want to, but Eric's discovery tells us something about him. His discovery was the first for Eastern Ontario, a new northern limit, and over 300 kilometers away from any other occurrence. Also, while in Kempville, Eric worked at the Eastern Ontario Biodiversity Museum, studying and teaching about plants and aquatic invertebrates as part of a biodiversity survey of the Drop River, which he led and described in a publication. Another Eastern Ontario naturalist says of Eric that he's an unusual field biologist with a broad and technical interest in many groups of organisms. It was a loss to the Ottawa area when he went to Peterborough. The same person says, we know very little about Eric, perhaps because he would prefer to discuss the behavior of rusty crayfish than talk about himself. 12 years ago, Eric was inspired to study mosses and liverworts by Jennifer Doubt, and he's been collecting them ever since then. He's currently a botanist in the Species at Risk branch of the Ontario Ministry of Natural Resources, Parks and Conservation, where he provides scientific advice on endangered and threatened plant species, including mosses. Now, Jennifer. Jennifer grew up in Deep River, Ontario, along the Ottawa River, and studied at the University of Guelph, and then went out west to the University of Alberta. She lived and worked in Western Canada for 10 years before returning to the East to take up her current role at the Museum of Nature in Ottawa. Jennifer has collected and documented mosses and liverworts for over 25 years, along with other dedicated staff, students, and volunteers that make up the Canadian Museum of Nature Botany team. She cares for a busy growing archive of over 1 million preserved plants and lichens, and shares the collections far and wide for research and education. Jennifer has sat on the committee on the status of endangered wildlife in Canada, providing expertise on moss species, and may still be active with COSEWIC. She's presently the treasurer of the Field Botanists of Ontario, and she's helped us with our last 2 BioBlitzes, actually visiting Maple Cross this past June, and previously identifying moss specimens that we'd sent her from the Hudson Rose property. We're hoping that both Jennifer and Eric may be able to join us at the Macaulay Mountain BioBlitz this June. And Jennifer is a real trooper, because even knowing that she would be in BC at a conference, she still agreed to give us this talk. So without further ado, I will turn the floor over to Jennifer and Eric so that we can all learn about bryophytes.
Great, thank you, Sheila. So Jennifer, do you want to try to share your screen? And actually, Marjorie put in the chat to remind people that if you do turn off your video camera, you'll still be able to see the screen on your computer. It's just people won't see your face, and it means less bandwidth that you're using. Okay, great. Would you like to try to do that, Jennifer?
Sure, I will.
Okay,
I also want to say thank you for the introduction. It was more fulsome than it was the last time I saw it. So it's wonderful. Thank you to whoever has filled in those details. I am the secretary of the field botanists, and somebody much more capable is in the role.
Oh, sorry. Oops.
No, it's all good. Anyway, so I'm going to actually turn off my video for the presentation, and I will now try to share my screen. And hopefully someone can confirm that.
Yes, we see it. It says, yes, we can see it, Jennifer. That's great. Thanks.
Okay. Eric, did you have any words of introduction after that? Or would you like me to begin?
Uh-oh.
Okay. I will begin, and hopefully Eric is still there. As Sheila mentioned, I became involved in Prince Edward County bryology most recently when Paul Catling passed some samples to me from the Hudson Rose property BioBlitz in 2019. And he said that he could collect some more if that was of interest, and it might be a little bit sad, but that's the kind of offer that I find difficult to resist. So he did bring more specimens back, and I enjoyed identifying them through the pandemic. And what impressed me the most about that whole experience was how professional and thorough the report was at the end of that. It was very satisfying to see the information formatted and distributed so well. So I was super excited to join the BioBlitz in person at the Maple Cross property, and so I had a good day at Maple Cross. I've been to the county as well to work on some rare species, which we might talk about later. But overall, I confess that I haven't spent a lot of time on bryophytes of Prince Edward County, although I've been working on bryophytes for quite a long time. Some of the knowledge that we'll share today is also from the Herbarium, the National Herbarium of Canada, where I work. And so that's where a good proportion of the specimens that are collected in the county, at least those that are on the public record, are deposited. Today, we hope that you'll enjoy this introduction to bryophytes. I, after all of these years, still feel like I'm only scratching the surface. So I know that for those who are kind of dipping their toe in it for the first time, it might seem like a lot. And I'd like to reassure you that we'll be really delighted if you leave simply inspired to take a closer look the next time you're outside. And if you are also inspired to send photos or stories about what you find, then that'll be an awesome bonus for us. Okay, so first up is, what is a bryophyte? A lot of people will just say moss, but there are actually 3 groups that make up the bryophytes: the mosses, the liverworts, and hornworts. Here are some photos with mosses here on the left and liverworts on the right, but we'll get into the details of those differences later. And then we have the third kind of bryophyte, which is the hornworts, and it's pretty rare. As far as I'm aware, no hornworts have been found in the county so far. In the entire province, there are a maximum of 5 hornwort taxa, and they're also ephemeral. They only show up at certain times of year and disappear again. So it's pretty exciting when you find a hornwort, and hopefully someone here will be the first one to find a county record for one of these creatures. The mosses are by far the most common of the 3 groups that make up the bryophytes. And when other organisms get confused with bryophytes, it's often because they have the word moss in their name. These are some names that might be familiar, things that are called moss, but in fact, only one of them is a moss in the true sense. So why don't we take a look at some of these and try to sort it out. I wasn't actually aware of Irish moss until someone asked me about it. It's an ingredient in some desserts and soft drinks. Apparently it can be used to clarify beer, but it's not a moss, it's a seaweed. Really, most seaweeds look pretty different from mosses, and in fact, mosses are not very tolerant of salt. There are a couple of species that can grow close to, like, are associated with ocean spray, so they grow on the land, but where the waves might toss up some salt spray. And there are some specialist mosses on salt flats. But if something was plucked out of the ocean, it's almost certainly not a moss. And we have Scotch moss, which is a ground cover. It's a popular ground cover. It looks kind of fuzzy and green, and from a distance, it looks kind of like moss, or it must have reminded somebody of moss at some point. It's often in rock gardens, so that might be another reason why. But here, the real key is the flowers. Mosses, as we'll discuss in a bit, disperse as tiny single-celled spores rather than as seeds, and they're not pollinated. So if you see flowers, you can be confident that you're not looking at a moss. But speaking of that, with some of the limestone habitats in the county, you might have run across stonecrop before. Maybe the people who posted these observations to iNaturalist are here, and if so, thank you so much. They're great photos. When a stonecrop is blooming, like in the top photo here, there's really no problem because you can see the flowers and you can rule out the fact that it's not a moss right away. But if it's not blooming, as in this picture on the bottom, it can trick you for a little bit. Okay, reindeer moss is pretty well known, maybe because there are blankets of it in certain, mostly northern parts of the world, and because it's used for crafts and decorating. Here you can see it's sold as decoration moss and super moss, and yet it's not moss at all. It's lichen, and I heard as we were getting started today that Troy McMullen has been here several times to give you the lowdown on lichen, which is a partnership between algae and fungi and other microorganisms. Is somebody trying to speak?
Please turn off your microphones if you have them on. Thank you very much. Sorry about that.
Thanks, sorry for my confusion. So lichens are a partnership between algae and fungi and other microorganisms, whereas mosses are plants. So lichens and mosses are not even in the same kingdom. They look really similar in size and from a distance in general appearance. So that's why when you encounter a lichen, you have to look up more closely. In this collage, we have a few different kinds of mosses. It's true that they vary in color. There's even a red one down in the lower left-hand corner, but for the most part, mosses are green. Here's a tree trunk where you could expect to see both mosses and lichens, but in this case, the growth you can see is green. And then when you zoom in, and here I mean zooming right in, like you have to look really close, you can see that they have actually plant-like shape. They have leaves that are arranged around a stem. And lichens, on the other hand, are mostly not green, especially when they're dry. The green color of the algae part of the lichen can sometimes shine through when the lichens are wet, but lichens are often white or yellow or orange or black or red or gray, and sometimes they are green. But when you zoom in really close, you won't see leaves and stems. This is a clump of moss and lichen growing together on an alvar. This one's on Manitoulin Island, not in Prince Edward County. But it's an opportunity to check out the difference between the lichen in the middle, which is kind of gray and white and kind of an irregular shape. And it's surrounded by mosses, which are green and have little leaves arranged around stems. It can be kind of fun to quiz yourself on a walk, kind of look at a clump from a distance and take a guess as to whether you think it's a moss or a lichen. And then when you get up close and get out your hand lens or your magnifier, see if you agree with that guess when you have that second look. Of course, just when you think you have it all figured out, you'll find a thalloid liverwort. And that's when things, they start to get harder to explain and take more experience. So we should get together again for part 2 of this introduction at that point. Some lichens are very green when they're damp, as I was saying. So this on the right-hand side is a lichen. You can still see that the back of this lichen that's kind of showing around the edges is white. So that's a bit of a clue that it's a lichen. And here on the left is a liverwort called Marcantia. You might have seen Marcantia, especially in greenhouses. It's one of the greenhouse pests, if you want to see it that way. And this liverwort, unlike the mosses that we've been discussing so far, they don't have a stem and leaves. They don't have that kind of arrangement or body format. And so you can see that these are quite similar from a distance. There are 2 forms of liverworts. On the right, we have the leafy liverworts, and they have leaves and stems like the mosses that we've been discussing. And then we have the thalloid liverworts like the Marcantia here on the left. Just to make things worse, when you see a lichen with this form, they call it a leafy lichen, and our leafy liverworts are not this form at all. This is a thalloid liverwort. Like I say, something that you could go down a rabbit hole on at a later time. Thalloid liverworts are fun because you see them less often. So they're the least common, I would say, of the groups that we've seen so far, but they're so beautiful. And they're bryophytes, so they're part of our presentation today. So that was reindeer moss. Now we're on to club moss. But once again, these are not true mosses. For one thing, these examples are larger than most mosses. They are spore dispersed like ferns are, but like ferns and flowers and trees, they have root systems and architecture inside the plants for moving water and nutrients from below the soil to the above ground plants parts. And mosses don't have that. which is why you might sometimes hear them referred to as non-vascular plants. So if the plant is rooted, fairly strongly, and if it's large enough that you could grasp it and pull to test that criterion, then the chances are pretty good that you're not dealing with the moss. I know this is getting kind of repetitive, but I also wanted to mention spike moss. It's another spore dispersed vascular plant. However, it's super, super small here. It's, really similar in size to mosses, and it's another one that fools a lot of people. You're in excellent company if it fools you, and one to practice on when you're bored of testing the difference between lichens and bryophytes. And finally, here's peat moss. After all of the tips we've been discussing about how recognizing moss, this heap of brown fibers from the garden center is definitely not moss, right? But actually, this is the one thing that is moss. When it was living, it looked like this, with green color and with leaves arranged around branches and stems. And it's a whole different discussion, but when you get a whole lot of sphagnum peat moss growing in a truly fabulous place called the peatland for thousands or tens of thousands of years, it accumulates as peat. And then by mining the peat, the garden center is supplied with the brown fiber that we're all familiar with. In a little bit, Eric's going to introduce you to some amazing mosses in Prince Edward County, and he wanted to make sure you were familiar with the main parts of a moss, which helps in differentiating one species from the next. So here are the parts that I'll touch on. But I can't emphasize enough how important it is to look in closely. So when we consult a field guide to identify a tree, we're often directed to features like the shape of the tree, the shape of the leaf, details of the margins of the leaves, and things like that. And for bryophytes, it's really not any different. There's no mystery if you're familiar with identifying vascular plants. You look at patterns in the shapes of the plants and the shapes of the leaves and details of the margins of the leaves and things like that. But in order to see those things, you really, really have to look closely. Like it means getting your face right next to the moss and to setting aside appearances and carrying around your neck a magnifier, like a 10 or 20 times hand lens, which I know that many field naturalists already have. And if you have your hand lens, you should be able to pick out these moss parts pretty easily. At the top, we could go from the top to the bottom. At the top, there's the capsule where the spores form and where the spores are released. And then the stalk that connects the capsule to the leafy part of the plant is called the seta. And these two top parts are sometimes present and sometimes not present. It depends on what stage of the moss life cycle you happen to catch the sample in. But almost always present will be the leaves and stem and rhizoids. So the bottom parts of this diagram are pretty much always there when you notice a moss on the ground and pick it up. Here are just a few of the forms that capsules can take. But the main point I wanted to show you was just how different these ones look. And this is just a small sampling of many. In this case, with your hand lens again, so holding it right up to your eye with a high power magnifier, you can see that the end of this capsule is covered by kind of a drum head with slits around the outside where the spores will sift out into the air. The seta can be straight. It's usually straight, but it could be short or long. It could be so short that the capsule is just sitting on top of the leaves, or it could be longer than 10 centimeters. And in this case, the seta is very curly. It can also be smooth or rough. So those are features that, that help when you're trying to figure out what you're looking at. And now these are some leafy stems. They can be wide spreading, or they can be tight to the stem. They can be very straight, or they can be wavy. They can be long and narrow, like the two examples on the top, or they can be wider in comparison to their length, like the ones down below. And here also you can see, as compared with the straighter leaves, some of them are curled over to one side. So again, there's just there's a huge and beautiful diversity of forms of bryophytes, but from a distance it's hard to appreciate that and to use that information. You really, it pays to slow down and take your hand lens and take a look. And for this slide, I just wanted to make the point that this is the same species here. This wide spreading leaves on the lower right is just the same moss as the one in the upper left, but it's rained in between. So you have the chance to get to know all the mosses twice, once when it's dry out and once after rain. So that's pretty much the limit of what you can see with your hand lens, and you can see a lot more with a microscope, and there's a whole world to discover once you go to that extent. And what we're going to share with you today does not rely on microscopes, but with your lens, you can easily see, 25 species without traveling more than 10 meters down the trail. So it sets you up for a lot of diversity, a lot of discovery in a short amount of time, a small area. And there's lots of opportunity as well, since there are relatively few people studying bryophytes, to add something new to the county list. And Eric, are you back?
Yes, I'm here and unmuted.
Okay, super.
Do you want me to go from here? Is that?
That's what I was expecting, but would you like me to skip ahead to the species?
Sure. If we're,
Okay.
okay. That's
Sure.
all right. We'll go there then with some common mosses. This is a selection of species accounts that we pulled from from a list of Prince Edward County species that we compiled in preparation for this presentation. And it is somewhat a random, or perhaps a better word would be a capricious selection of species, in the sense that we could have taken a completely different group of common mosses and presented them. And the purpose here isn't really to be comprehensive in any sense, but just to familiarize you with a few species that you may be able to encounter and recognize in a relatively short period of time. So we'll start off with Pseudoanomodon attenuatus. It's a pleurocarpus moss. That means, well, I'll explain the dichotomy between pleurocarpus and acrocarpus. We'll see some of the latter a little later on in this selection. This is a distinction that it's somewhat like the distinction between a tree and a herb in application to vascular plants. It's not a taxonomic distinction. It's purely it purely refers to the growth form of the moss. So acrocarpous mosses terminate on their main stem and main branches with the reproductive part. If it's a female or bisexual plant, therefore it will have sporophytes developing from the ends of those branches or stems, while the pleurocarpous moss doesn't terminate in that way. It will have sporophytes, for example, developing anywhere along the stem from a short branch, and it will continue to grow in an indeterminate way beyond the sporophyte or other reproductive part. So this is it's sort of a yellowish green moss. It's what's distinctive about it, as you can see in this photograph, are the long attenuate tips to the branches, which are sort of regularly positioned along the main stem. It grows generally on the bases of trees, sometimes on rocks, and it forms these very thick or dense mats that, once you're familiar with this species, can be quite distinctive, and you can often recognize them from a distance. We'll go to the next slide. That's okay. This is another very common moss, Ceratodon purpureus. It is an acrocarpous species. So it's, as you can see in the top left picture, there are lots of sporophytes, and they're sort of right at the end of the leafy part of the moss extending into the air. It's quite variable in color, and it can sometimes be quite deceptive, but typically it's sort of a purplish or brownish green, and it has these very distinctive red setae. You'll probably encounter this species along, a gravel trail or even along some pavement, and you'll notice those a purple setae before you notice anything else. Next slide. Then we have Climacium dendroides, pleurocarpus, but looks very much like a tree. That's where the epithet comes from, dendroides. If you look at the lower left picture, you can really see the sort of tree-like form with the unleaved part down here, or unbranched part down here, and then this looking like the crown of a tree. It tends to be a dark yellowish green, usually on organic soil, often at moist to wet locations. So you'll frequently find it in wetlands, tree swamps, for example. Next slide, please. Yes, and then there's Centrichia ruralis. This is another acrocarpous species, green with sort of rusty or orangey secondary coloration. It has very distinctive long white awns on reflex leaves that become very twisted when dry. So here you see the distinction that Jennifer was mentioning between a moist or wettened plant. The leaves are widely spreading, and you can see the white awns very distinctly. And in the picture beside it is a dry, desiccated plant. The leaves are tightly oppressed and sort of twisted against the stem. And even the awns are less noticeable. They're sort of in a tight little twisted package up at the top of the plant. This plant occurs on sand, gravel, even on old pavement, and you'll find it along some of the shorelines, like shoreline cliffs in Prince Edward County. Next slide. Now we have our first liverwort. And as Jennifer mentioned, here we also have a dichotomy of the same kind. We have foliose and thallose liverworts, although this does actually map broadly to taxonomic division. So our first one is Lofocolia heterophylla, so named because it has very variable leaves. The typical form that you'll probably notice first when you get to know this liverwort is the one with large bilobed leaves. It's a light green plant. It tends to have the leaves flattened against the substrate on which it's growing. And it has a very distinctive triangular perianths. That's, I'm assuming you can all see my cursor. Hopefully that's is the case. Yes, down here. That is sort of the perianth is a modified or fused leaf or set of leaves that surrounds the reproductive part of the liverwort, the female part of the plant that produces a sporophyte. So this species of liverwort typically occurs on woody debris that can be sticks or small branches on a forest floor, or on a large rotting log, or sometimes even on soil. Next slide, please. Another very common foliose or leafy liverwort, Parella gladophyla. Tends to be yellow-green with rounded sort of scale-like leaves. You can see, I guess it's probably most visible in the picture to the right where the plants are sort of dry. You can see the overlapping scale-like pattern of the leaves in that photograph. Underneath the leaves, there are lobules. It's sort of a almost like a smaller leaf growing on the bottom of the plant, but it's not the underleaf, which is typical or found in many liverwort species. Very characteristic is the growth form that you see in the top left picture. When plants are well hydrated, they tend to have this sort of ascending growth form. You see them stretching off the surface of the tree on which they're growing here. And these are typically found on trees or shaded rock outcrops in forested areas. Next slide, please. So one more common liverwort of the foliose or leafy kind. This is Radula complanata. It's green, round-leaved, also scale-like leaved species. It has marginal gemmae, which you'll need some magnification to see, but a 10 or 20 times hand length should suffice. And those gemmae will look like just little particles on the formed on the edges of the leaves when you look very closely. It also grows on the trees, logs, rocks, and soil, so it's quite general with respect to the sort of substrate that it inhabits. In moist, shady areas, it can be very abundant and will completely cover fallen logs in the bases of trees. Next slide, please. Then we have an exciting thallose liverwort. This is a Climacium salbrosum. It's a very large liverwort, shiny green with this distinctive reticulate pattern on the upper surface of the plant. It looks sort of like snake skin or skin of some species of lizard. It's very distinctive. When you see it, you'll probably be able to recognize it right away. And it tends to grow on basic soil or rocks, usually in a moist environment, often near running water. And then now that we've talked about a few common species, we'll go over a few of the rare species that have been documented in Prince Edward County, starting with this one. Bryophytes gemmiparum. It's an Aquaculture moss species, green to yellow-green. Probably the most recognizable field characteristic is that it has very concave upper leaves that form a sort of a distinctive bud at the top of the plant, and you can sort of see those in the picture, the accompanying picture, which is from Prince Edward County, the southeast point of the county. And the species is mostly on basic soil over limestone. Next slide. So this is another Aquaculture species, Orthotrichum pallens, green with very narrowly obtuse leaves. And you can see the sporophyte on these plants. They're emergent. That means they're sort of extending beyond the leaves. They're not, this seta isn't visible beyond the leaves, but the capsule is. And it's an epiphyte. It usually grows on trees. This is one of Jennifer's discoveries. So I don't know if she wants to tell you where it is in the county, but if not, we'll just go to the next slide.
Okay.
And then another wonderful discovery that Jennifer made. This was from McMahon Bluff, I believe. Dramadia propens, a pleurocarpus moss, sort of green or brownish green. It has these prostrate stems that grow along the substrate on which the plant is, to which the plant is anchored. But the branches are erect and they have this sort of distinctive pattern resulting from very closely compact erect branches. It's a monotypic genus, Dramadia, so this is the only species we have in Ontario, and it's also an epiphyte, usually growing on trees.
Maybe I'll just say one thing about how special this was for me to see at McMahon Bluff. I had never actually seen it in the field before, and one of the reasons is that it had not been collected in Ontario for about 100 years. So it was a real thrill. I didn't know in the field what it was. I knew it was different, and it was kind of drawing lines on the tree trunk that it was growing on, just like you can see around the margins of this colony in the picture. But yeah, it was a thrill for me to see it, and also a feather in the cap of McMahon Bluff that the species is there.
And I think this is our last rarity, Schistidium crescipulum, an Aquaculture species, which is sort of this dull olive green or brownish. On this species, the sporophytes are immersed, meaning they're sort of set deep in amongst the leaves, both the seta and the capsule. It's a species that grows on basic rock, including on built structures such as stone walls and rock piles. And this one was actually found on a rock pile on Timber Island Nature Reserve. And there aren't, well, I haven't found any previous records of it from Ontario, but there are some records from, there are a few records from Quebec, and I think there are quite a number of records actually from where Jennifer is right now. You're probably just surrounded by representatives of this species where you sit at the moment. But it's quite common in Europe, and I suspect given the sorts of substrates on which it occurs, it might be more common in Ontario and probably Quebec too than would be suggested by the current state of knowledge. So next slide. And then we have one species to highlight that is considered to be still to be found in Prince Edward County, if we can be so hopeful. And I think Jennifer is going to talk about that since she's been doing some advocacy for the detection of this species in Ontario.
Yeah, this moss has been my quarry for the past few years. It's another moss that was, it was present in the 1800s. I'll show you a little graph in a second, and it then was not found for more than 100 years before it finally was rediscovered in Quebec. And since then, there have been a few more populations discovered in Ontario as well. But I'm really excited for many reasons to go to the BioBlitz in Prince Edward County this year, but one of them is that maybe this plant is there if you have old trees and shady rocks. It's called fan moss, and the scientific name is there as well. But if you go on iNaturalist, it's called fan moss. You can see that it has, similar to the porella that Eric was showing, it sweeps out from the vertical substrate that it's on. So the rock or the tree, the branches curl away and a little bit up at the tips. And it has these cool orange kind of capsules mixed in the foliage, which sets it apart from some of the plants that look similar. This is what it looks like in the rain and on a dry day, because it looks quite different in those two situations. And this is the distribution. So where you can see stars are populations that are confirmed extant today. There's another one in Frontenac Park, so sort of in this area. And then the ones without the stars, those are collections that were made in the 1800s. So, Prince Edward County is right in the zone here, and it was historically collected in Belleville, as many plants, rare plants were, because that's where John McCown lived. So why not in Prince Edward County? That would be amazing to find it there. And so maybe I'm just going to go back so you can take another look at that slide. If you happen to be out just walking around in nature and you notice something that's shaggy, some shaggy moss on the side of a tree or a rock, and you see these orange capsules interspersed, please take a picture and take a waypoint and let me know. I would be so grateful to be able to find more. And the reason why, course, it's fun to look for these things, but it's also a species that's about to be assessed by COSEWIC, the Committee on the Status of Endangered Wildlife in Canada. And so getting information about this species now is really important to an accurate assessment and, you know, any protection that is due to make sure that it remains a Canadian species going forward. I also was just going to say a couple of things about the bryology of the county. There haven't been that many people who have been collecting there. I'll mention them in a second, but so far as we could tell by looking at data sources online in the herbarium and consulting our own collections and those of people that we know in Ontario who are active in collecting in this region, there are at least 102 mosses known so far and 16 liverworts. Hepatic is just another word for liverwort. But this is pretty much impossible that there are only 102 mosses and 16 liverworts in the county. So it means that anytime that you go out and happen to collect, you have a high chance of finding something new and expanding this list and contributing to the knowledge and conservation of bryophytes in the county and in the province. This is just a sample from the herbarium where I work, and it was collected by Jean Gagnon. He was working for the Ministry of Natural Resources at that time, and he's one of the people who's responsible for many existing county records collected over here in the east part of the county. These two people, Robert Ireland and John McCown, were both affiliated with the Museum of Nature and its former incarnations, and also have contributed records to the county list of bryophytes. Both of them were very prolific collectors and very knowledgeable about plants. Quite a few Ontario government employees have visited the county and added records. This is a rare liverwort that Mike Oldham had found. And here are some other people. Bill Stewart, incidentally, was in the county because he was doing a survey of bryophytes on gravestones in Ontario. I think that headstones are kept cleaner than they were at the time, but it's a really interesting survey and wide-ranging in the province. You can find different species on monuments than you might in the surrounding habitat if the rock is not typical of the region. So there's probably extra diversity in the county because of Bill Stewart working on cemetery mosses. And of course, if you go out and take an interest, you have, as I say, a good chance of finding something new. Back to Eric.
Okay, so you may recall from the beginning, I think our last section was how to get started, and to that end, I'm going to go over some of the equipment and techniques that you might use to start to find mosses and liverworts and perhaps even a hornwort in Prince Edward counties. So we'll go over field collection, specimen preservation, which really comes at the end, but we're going to do that first and then talk a little bit about specimen diagnosis, what references you need, and using iNaturalist. So collecting locations, that of course depends on what you're looking for. For the beginner, really almost anywhere that you may want to explore, whether it's a forest or lakeshore, cliffs, wetlands, alvars. All of those are potential areas where you will find mosses and liverworts, and will likely find some things that that haven't been documented in the county previously. What sort of equipment do you want to bring with you? So I'll go over what's in the picture. What these little brown paper bags, I suppose they're sandwich bags, are what's typically used to hold a specimen that you collect in the field, and is used as sort of a temporary repository while that specimen dries out once you bring it home. You'll also want to have a notebook in order to write down some information about where the specimen came from. You can also write that, of course, on your paper bag. But for a more permanent record, you may want to retain it in a notebook. You ideally should have a GPS unit so that you can take a waypoint and have coordinates associated with your collection. You may need a knife in order to remove a section of a moss colony from a substrate, particularly if it's growing on the bark of a tree or something like that. And as Jennifer mentioned, an eyepiece so that you can look at what you're collecting before you collect it, and have some idea of what range of diversity is represented in the area from which you're collecting. Now, as I said, we'll jump to the end and look at what we want to have as a final specimen. So this is actually a couple of packets holding the Bryum gemmiparum specimens from Prince Edward County. You can see in these packets are made of non-acid herbarium paper. They can be purchased at a herbarium supply store, and they're specifically designed for bryophytes and lichen specimens. And then if you do start to collect a lot of mosses and liverworts, as you become more and more enthusiastic, something which happens inevitably, you may want to start your home herbarium. For that purpose, you can store these in a simple plastic container like this. There are special boxes manufactured for bryophyte specimens, but I think they're probably quite expensive. Jennifer would have a better sense of what the costs are, but I think these plastic containers are quite satisfactory for this purpose, particularly if you can keep some sort of silica desiccant in with the specimens to ensure that moisture isn't getting trapped in the plastic container. Next slide. For the diagnosis of specimens, there are some basic tools that you will want to have. Forceps, an eyedropper to rehydrate a section of moss or liverwort in order to examine it more closely once you're at home and you have your references at hand. On the left is a reticle. That's just a device for measuring leaves and stems or entire plants under a microscope, if you have a microscope at your disposal. And similarly, you may need a razor to dissect plants or parts thereof. So not everyone will want to take the step of using a microscope, but ideally that is what you need to fully identify mosses and liverworts to species level. And you'll need both a stereo microscope as depicted here and a compound scope as depicted here. The stereoscope is used to remove parts of the plant that need to be examined, and the compound scope is used to examine those parts on a slide at much higher levels of magnification. And I think we have some pictures here of micrographs. This is sort of the level of detail that you can observe under a compound microscope. You can see all the cells of the leaf of a moss, this central midvein or costa, how long it is relative to the rest of the leaf. You can see the different cells along the margins. Here you can see the toothing of the leaves in great detail, and you can measure cell lengths and the lengths of leaves and things like that, which is often relevant to determining what the species is. And here we have some nice high magnification pictures of cells of sphagnum, again showing the level of detail that you can see under a microscope, and often a level of detail that's necessary in order to identify the plant species. Here also some more pictures of the cells. I think these are all from the leaves or leaf lamina. And in this picture, the lower right picture, you can see little papillae on the surface of the cells. And I don't have a scanning electron microscope. I don't think most people will go to that length, but of course, if you're affiliated with an institution that has such equipment, you can really obtain beautifully detailed three-dimensional images of the parts of moss. And then references. So what sort of books do you want to have in order to build your home bryology library and be able to identify mosses, liverworts, and hornworts at home? So books about mosses, there are a number of tomes that are very useful for our part of the world. On the left is Crum and Anderson's 1981 Mosses of Eastern North America in 2 volumes. It's probably the most comprehensive reference for Eastern North America on mosses. It's maybe a little dated in terms of taxonomy, but can still it's still an invaluable resource for identifying many of the species that you'll encounter. And then there are a few other examples here. I'll jump to almost to the end. Mosses of the Great Lakes Forest is still quite a popular resource for people who are learning bryophytes, or specifically mosses. Robert Ireland, who you saw in an earlier slide, his moss flora of the maritime provinces. I think it's hard to obtain in book form, but it is available on the Biodiversity Natural Heritage Library website. And then Maine Mosses also by Bruce Allen. For liverworts, a really wonderful book to start with if you're get very interested in liverworts. In Ontario is An Enthusiast's Guide to the Liverworts and Hornworts of Ontario by Linda Lee and Joan Crow. Linda Lee works a lot with Jennifer at the Canadian Museum of Nature, and is extremely knowledgeable about liverworts, particularly Ontario species. So that's a handy, almost pocket-sized guide specific to our part of the world, with some easy-to-use keys and illustrations of the vast majority of species that you're likely to encounter. And then if you really become dedicated to the cause, Schuster's The Hepatica and Anthocerote of North America, east of the 100th meridian, I believe. That's a 6-volume set. Most of those volumes are quite difficult to obtain. They're no longer in print, but if you can find copies of them, they are a really comprehensive and in-depth accounting of Eastern North American liverworts. And of course, there is the internet. The image I've put up here is for the flora of North America at www.eflores.org. Volumes 27 and 28 of the Flora of North America are dedicated to mosses. Volume 29 has not yet been published, but there are some draft or in preparation accounts available online for the liverworts. And this is a very good resource to get started on if you're interested in identifying mosses and eventually liverworts, particularly if you know the genus or perhaps the family in which to which your specimen belongs. You'll be able to then select a specific key and work out what species you have. Oh, and I should mention, just for online resources also, there are some websites that have very good illustrations of or photographic images of mosses and liverworts, both in the field and micrographs. I think one of the ones I'd really recommend is the British Bryological Society website. It has a lot of very good images that will be helpful. And now iNaturalist. So another way to both to document your observations and also to receive well-informed input on identifications is iNaturalist. Probably many of you, if not all of you, have iNaturalist accounts, so you can post observations of mosses and liverworts that you find, and often someone will offer an identification. Perhaps Jennifer. So what sort of information do you want to post, particularly if you have no microscope? It's very informative to have a habitat photograph, something that will give an indication of where the plant or plants were growing. A photograph of the patch or colony from the top-down view and the side view, and then close-up pictures of leaves, and if present, the capsule and seta. And so here's an example for Fiscomitrium piriforme with on the left, sort of a habitat picture. You can see the colony in the forefront, but you get that broader view showing the sort of environment in which these mosses were growing. And then you get on the right, a closer picture giving more details on what the patch looks like. And I think we have in the next slide an even closer one, yes. So down here, a close macro photograph, which really allows you to get a sense of what the capsules look like, what the shape is, and you can even see some of the leaves of the moss at the base of the capsules. And these, I think, are just some more habitat pictures, sort of information that's useful for making a determination.
Yeah, and I also dropped them in here because it's a reminder that the spring and the fall are really excellent times to look for bryophytes. You know, things are starting to leaf out now, but when the snow goes away, you can see all of the mosses starting to glow and to, you know, respond to the sunlight that they have access to before the leaves come out on the trees and before the understory fills in. And you don't have a lot of distracting flowers out there as well. So if you want to extend your botany season, then early spring and late fall are perfect times to get a good introduction to mosses wherever you happen to call home. Oh, and so I just wanted to mention the herbarium again before we conclude. This is the team that we have right now. Most of these people are students who are in the herbarium for the term. And it's in Gatineau, and it's available to everybody. So if there are plants or lichens or bryophytes that you're interested to see, or if there are specimens that you'd like to access for a project so that you can get to know them better or know where they were collected and things like that, then you just need to be in touch and we'll set up an appointment for you to come in and help you find what you need. We also have an open house, especially now that the pandemic is starting to ease off. Hopefully we'll start to have our open house again, which happens in October, and then we open up the doors not only of the herbarium collection but also to the other collections in the museum, so paleobiology and zoology, vertebrates and invertebrates, as well as mineralogy. So please be in touch or come by. We'd love to show you the collection. And thank you so much for your attention.
This is thank you. We've got a few questions here from Heather of Fiddlehead Farms. Are liverwort reproductive structures always like those in the photo and unlike those of lichens?
Yes, I wonder if I can go back to that slide to just make sure we're on the same page.
Just in general is fine.
Sure. Well, the reproductive structures in short, I'm gonna, maybe I won't try to find it after all. The reproductive structures in short are not the same between lichens and bryophytes. So it is a good clue if they happen to be present. The picture of the Marcantia near the beginning of the talk, there were some cups on the surface of the liverwort, as well as some sort of umbrellas kind of cartoon umbrellas that were attached at the back, and those would not be present on a lichen. But if it's not the right time of development, when you run into the Marcantia, then they won't be there and you won't have those clues.
Okay, Heather. And another one from Heather. How long does it take to rehydrate in the field? Can you splash with a water bottle?
Did you want to take that one, Eric?
Yes, you can. Yeah, it doesn't take long. Probably a few seconds, maybe a minute. So yes, you can do that and get both the wet and dry impression of the plant while you're still in the field.
Thanks. So if they don't have roots, how do bryophytes get their nutrients?
That's a terrific question. They're basically excellent sponges. So I guess one of the reasons that you can get such a beautiful view of moss leaves with a microscope is that they're just one cell layer thick. Like the top of the leaf is the top of the cell and the bottom of the leaf is the bottom of the cell. So you can see right through it, but it also means that the cells are pretty much all exposed to ambient moisture, and the water and nutrients are just absorbed directly into all of the different cells of the plants. And the other side of that is that then without roots, the moisture just leaves again. You know, when the weather turns dry, then they dry out and then they can't grow again until they have more rain or dew or other water.
Actually, something I just thought of, how long will these plants live? Do they come back in the same place year after year after year?
Well, my colleague in Alberta went looking, he got out Thomas Drummond's diary. I'm not sure if you've heard of Thomas Drummond, but he was part of one of the Franklin expedition members in the early 1800s. And, you know, it was part of a different leg than the one that Franklin took of that trip. And he discovered a moss that ended up being rare today. So my colleague looked at Drummond's diary to try and figure out where Drummond had been so that he could go and rediscover the population. So lo and behold, in, I don't know, it might have been 2001, using information about where Drummond was camped out, he went back and found colonies of that same rare moss in that same place. Was it the same plants? Probably not, but they're, you know, probably clones or descendants of those original plants. The colony was there, even if the plants are not the same. They don't actually, in the winter, they don't disappear. That's why they're so quick to start up in the spring, because they're just under the snow. They're sort of in suspended animation, and as soon as the snow melts and the water turns liquid again, they're still there and they're ready to go.
Oh, thanks. That's fascinating. You know, you might have answered this one already, or Eric might have, but it was from earlier, a question from earlier. How do you store your samples at the museum? Is there more to add than what we've seen in the slides?
It's very similar to what Eric showed. Our boxes are different, just because the cabinets are a certain standard size and shape, and the shelves are a certain standard size and shape. So our boxes are a different shape, but everything's arranged kind of like a card catalog on their edge, the way that you saw in Eric's photo. And everything is, you know, we have 280,000 samples in our herbarium. So there's fairly rigorous system of organization so that we can find what we need when we're looking for it. You hopefully saw the cabinets in that picture. So inside some of those cabinets is just, you know, trays of carefully organized, lovingly preserved mosses.
That's amazing and wonderful. And last question so far, and again from Heather. So if anyone has a question, send it in quickly. When are liverwort and moss capsules most likely to form? Are they seasonal? Do they depend on the rains? Does vary by species or genus?
You have, did you want to take that one, Eric?
I'll start. I think they're not quite as strictly seasonal as say vascular plant flowering, which of course is variable itself. They're being late and early flowering species in the over the growing season. For mosses and liverworts, it varies depending on species. There are some mosses now producing sporophytes, or others that will probably not do so until later in the summer. Some species are known to produce their sporophytes in late winter, you know, in February or March, early March. Do you want to add to that, Jennifer?
Only to say that there's no, you're not going to miss the season because all the different species are going to produce sporophytes or not at different times of year. So that's kind of convenient, both because you're not going to miss the season, but also because if you return to the same patch, you know, in a few months, maybe you'll find it producing sporophytes if you didn't before.
Okay, thank you. So that's the end of the questions, but we've got two pieces of good information here. And another question. So from Rebecca Lamb, another great book, but not an ID guide, is Gathering Moss by Robin Wall Kimmerer. Don't know if you know that one or not.
And it really is an amazing book. It's a whole different view on mosses than we have presented here, and a very inspiring one. You know, if you're tempted to look at mosses more closely, I highly recommend it.
Okay, thank you. I think some of us have read Breeding Sweetgrass by the same author. And from Les and Anne to everyone, according to Mr. Google, mosses live typically 2 to 10 years. Lichens, 500 to 1,000, no, sorry, to 5,000 years. Wow. So thank you, Les and Anne and Mr. Google. And then from Rebecca again, how do you feel about moss milkshakes? Rebecca's a forager. So how do you feel about moss milkshakes?
So Rebecca, maybe you could clarify, do you mean in terms of growing mosses or actually consuming them?
No, that's a good question. I meant more in the way people think they try to propagate them by making the milkshake and growing it. Please don't drink a moss milkshake.
I've never had success with that technique. I've tried it for sure. At the museum, we have two buildings. We have the research and collection facility that I showed in that slide, and then we have an exhibit building in downtown Ottawa with a garden around the outside, which is loosely modeled on certain iconic landscapes of Canada. And we were hoping to grow some mosses in that space. So one of the techniques that we used was the moss milkshake technique to try and grow mosses on some of the rocks, and it failed miserably. The best way that we were able to grow moss in that garden, city kind of rocky landscape, was just collecting and drying and fragmenting mosses that would grow in a disturbed place, and then sprinkling them in kind of gravel where they could fall between the rocks and stay moist for a longer period before they grew. And they filled in quite a lot, just that way. On the other hand, when I was living and working in Alberta, we did manage to grow some moss on a rock surface, and it required somebody to go and mist it, you know, several times a day for several months before it really took. And it took, but it was a hard, dedicated job.
Okay, thank you. Well, I think that's probably it for the questions and
I'm gonna,
comments.
I
Actually,
think there's one question.
Jerry, let's see.
Oh, I
There's a
missed
question
one.
from Paul Catling with his hand up. Sorry about that.
Okay, Paul, go
Paul?
ahead.
Yes, thank you very much. I wonder, I think both of you would be interested in this question, so it's really directed to both of you. And that is, I think you've shown us some of the interesting rarities and distinctive features of mosses in Prince Edward County. And of course, we're used to significant flora and fauna in Prince Edward County. And it's very nice to know more about some of these things. But there are things that we recognize, or we have to admit, that we don't have in Prince Edward County now and then. Now, of course, they're more than compensated for by the rare things that we have and the really important things that we have. Nevertheless, I wondered if either of you or both of you might like to say just a little bit about some of the interesting mosses that occur elsewhere that we don't have. And I'm thinking particularly of the variety of mosses that occur in fast flowing rivers in which the county is a little deficient. And also mosses like sphagnum, which I think has a fascinating story, but you know, it's a story that is, I guess, mostly far to the north of the county. But would you mind, I think the people would be interested in that, and I just wondered if you'd mind telling us a little bit about those kinds of things that we don't have here.
Do you want me to start? I could say something about sphagnum is or the family Sphagnacea. It's a very interesting family of mosses in that it has the sporophytes are in many species quite showy. I think there was a picture of one on one of the slides earlier when you were showing the variety of sporophytes or capsules, Jennifer. These are mosses that typically grow on feces or dung, and they have these sort of showy sporophytes which attract flies that are initially attracted to the feces, and the flies are then induced to spread the spores similarly to the way that insect pollinators, I suppose, pollinate flowering plants. Yeah. Oh, there's is that Aplodon?
Yeah. So this is a picture from the Arctic, actually, and this is very old muskox.
Yeah.
Dung. And so, as Eric said, these this moss, these capsules attract insects. In other cases, the capsule actually looks like a little iridescent umbrella. They can be yellow or very showy, kind of hot pink purple. They look more like cartoon mushrooms sometimes than mosses. And they grow, a few years after the dung has appeared on the landscape, and after it decomposes, then this moss will have to have dispersed to a new spot with the help of insect air
So
mail.
our problem then in the county is that we're deficient in moose, is it, and bears and so on?
Yeah, I mean, that and the habitat that many dung mosses like is quite wet. So if you're in an area that happens to be very dry, you also wouldn't find this kind of moss. Now, in the herbarium, we have specimens of dung mosses from the Ottawa area from 100 or 150 years ago, and you would never find it around Ottawa now. But I'm not aware of any collections ever from Prince Edward County. Maybe
Thank you.
I will.
And
Sorry.
with respect to mosses that grow in rivers, what kind of diversity are we missing there?
Well, I mean, there are mosses that grow in flowing water, but you must have streams in Prince Edward County, even if there aren't rivers. And I expect that there are some aquatic species of flowing water in the county, even if there are no rivers. But what might be more important is the chemistry. So if you're not on the Canadian Shield and you don't have as many of the acidic rocks and acidic substrates, then you might not have, quite a large community of bryophytes that prefer that kind of environment. And one that came to mind when you asked about interesting species that don't occur in the county is called Goblin's Gold. It's the most amazing thing to see. It grows on, sand, grows in acidic soil, mineral soil, in the dark. So in the pits that form under roots, when trees fall over, there's kind of a cave that's formed where the roots come up. And this moss pretty much glows in the dark. I mean, it's not actually fluorescent, but it's so adept at catching the light and reflecting it that it looks like there's a light coming from underneath the tree roots. And yeah, so that's worth watching for as well, but I would be very, very surprised if it showed up in Prince Edward County.
Thank you.
Thank you.
Sorry, Paul, did you have more?
I actually have a lot more. Maybe, thank you for that offer, though. I may as well ask one more question now that you've let me in. And that is, what about the, whatever they call it, I've never been able to get this right, the inlier or the outlier at Ameliasburg, the huge area of granite, which is the southernmost granite that we have anywhere in Ontario. I'm just wondering what moss experts might think about that and about how interesting that might be, and whether or not we know very much about the mosses and liverworts on that landform at the present time. It's something we don't often, we know that it's there and we're interested in it, but it's not one of the places that we really identify as exceptional in terms of its biodiversity. but I'm just wondering whether it might be more exceptional based on the fact that it's the southernmost occurrence of anything like that in Canada, and also mosses, which require, some species of mosses, many species in fact, require acid granitic rocks. So
Yeah, unfortunately, or fortunately, the inlier is on private land, so we can't go up there and examine what's there. But you mentioned a name earlier. Is it Macomb?
McCown.
Jennifer? He was at his house at one point, Amy? Am I?
Well, Sheila can answer that, but he's one of the people that wrote about it and
Yes.
discovered it, I guess, and
Yeah.
at least, you know, documented what was there, according to
Yeah.
Mark Forget. Yeah.
Yes. So if anyone would have, if anyone has access to his books or research, that would be a good place to look for that. Sadly, we can't go and look now because it's on private land. But thank you for that. I'm going to close questioning down now because it's almost 8 o'clock, and Eric and Jennifer have been very generous with their time. From Lise here, Lise Bois, thank you for a very informative presentation. I will definitely pay more attention to mosses and lichens. And I would like to thank you too, on behalf of everyone, Jennifer and Eric, for a really fascinating look into what is to some of us an unknown miniature world. You can see that you've had a wide range of experiences and knowledge here on bryophytes, but I think it's been fascinating to all of us. And then at the end of my little handling of questions, I always say, or sometimes show, that we always have a gift for our speakers, because you both live quite far away, and because we hope you both will come to Macaulay Mountain and to the BioBlitz. We will either deliver your thank you gifts then or find another way. I'll turn this over to Amy now, but thank you, both of you. It's been amazing.
Thank you. It's been a real pleasure. Thank you very much for the opportunity.
Yeah, thanks very much. It's great to have some interest in bryophytes.
Thank you.
I'll concur with Lise. I got really excited too. This is a completely known world to me. So thank you both so much. I'm going to stop the recording. Okay.